Okay, guys, so my name is Mary Beth Panagos. I know some of you in this room, but some of you are fresh faces. I'm Director of Business Development for Panagos, but I'm actually here in a different capacity. So I started working with BRC in year one of Triangle Biotech Research Symposium, and we did the inaugural symposium together. So how many people were here in year one? Okay, no one that I see. Okay, some of them are already out in the lobby because I've run into many of them. But I'm really glad to have you guys back year over year. The idea behind this is, just as the Biotech Center announced, to have incredible collaborative minds in one room. This was set up as sort of an alternative to TED, which I had helped organize back in San Antonio. And I think we just didn't need the branding, we just needed the minds. So I'm really glad each of you are here. So I'm going to go ahead and get started right away. So of course this panel is on CRISPR. We'll be discussing this over the next hour. And I'd like to introduce the chair for today's panel is Dr. Barrangou. Now there's a lot of numbers associated with Dr. Barrangou. 9 years in industry, research and development, mergers and acquisitions. He's got 2 masters in bioengineering and food science. He's got a PhD in genomics and an MBA. So that's a significant amount of schooling, and now he's applying all of his skills and his passion at NC State. So, Dr. Barrangou?
Thank you for the introduction. I think it's my 3rd such meeting here. I've enjoyed them all in different ways. They're very colorful, and I'm going to use some of that schooling, arguably, to share some stories about CRISPR, just generally speaking, introduce the topic more than anything else, and echo some of the themes that we heard today. And the theme for the major conference today is convergence, and I can hardly think of a more converging field than CRISPR. I think it's more merging than converging because it's happening so fast. It's like the highway coming in from Durham or NC State this morning, right? And it really feels like the whole world is merging into the CRISPR space. I know it feels like this to me. I'm sure it feels like this to my fellow panelists here today. The second point, to echo some of what we heard this morning from Paul, is it takes a village. And in the case of CRISPR, I think it takes the whole world to build CRISPR, right? And I'm going to try to illustrate that to some extent for sure. And then one interesting item that I hope we'll discuss during the conversation part of the panel here today is the valley of death actually has not hit CRISPR whatsoever. And if anything else, it was a very quick, rapid segue from the innovative part and discovery part, arguably all the way to commercialization. And we've seen some commercialization in the biotech space occur actually within months of some of those proof of concept works coming out. I'm going to predict comfortably and confidently that it's going to take a lot more time to make it all the way through the FDA path, as we all know, but I hope that we get to discuss this today. So if we can have the slides, please, and get started. My objective here is really to give you a quick introduction of what CRISPR is, okay, so I can set the stage for some of my fellow experts here well, we have to represent NC State as much as we can, especially with 2 Duke guys on the panel, right? So I'm not going to waste too much time explaining what CRISPR is or how it works, but I feel compelled to say what it actually does in nature, because it's not just a genome editing tool. It is a genome editing tool. CRISPR equals genome editing, but actually it does a lot more than that, and natively in bacteria, it's an adaptive immune system. And CRISPRs look like this. I'm going to spare you the DNA details. It's clustered, regularly interspaced short palindromic repeats. CRISPR, it's a very catchy acronym. And essentially, it's a very weird array of DNA repeats that look like such, interspaced by what's called spacers. So you have the CRISPR repeats, you have the CRISPR spacers, and the whole system works as an adaptive immune system in bacteria, whereby you get infected by a virus. In this particular case, a bacteriophage. We'll hear about real viruses later today. And then a piece of the DNA gets hijacked and copied and pasted into the CRISPR array as a new CRISPR spacer. That's a vaccination event for the cell. Essentially, it's taking a genetic mugshot of the invading virus. So it's DNA encoded. And then once you are vaccinated, this whole CRISPR array will get transcribed and processed into small interfering RNAs, much like RNAi in some ways. Okay, there's some differences and commonalities. I'm not even going into that, but it's DNA encoded, RNA mediated, and those small RNAs that contain a portion of that spatial sequence here really target in a sequence-specific manner homologous sequences in viruses. And this by complementation, and essentially those guide RNAs or CRISPR guides will direct Cas nucleases, and the Cas genes are associated with the CRISPR array, for sequence-specific cleavage of that mugshot type of DNA. Okay, so DNA encoded, RNA mediated, DNA targeting immunity. Now, the real rise of CRISPR and the CRISPR craze we've all witnessed in the last couple of years has been fueled by genome editing type applications. And essentially what CRISPR has turned into is an RNA-guided DNA nuclease. It's a molecular scalpel used for DNA targeting and cleavage. And that molecular scalpel is very specific. It's only a two-component system. You need one protein, the infamous Cas9, but other Cas proteins do that. You need one single molecule to direct it, called the guide RNA, and then essentially you generate what's called a double-stranded break, a DSB. And when you cut DNA in a cell, the cell is typically not happy. One option is to die. Nobody likes to do that. So what the cell is going to actually do is going to try to repair that break and reattach that DNA using homologous directed repair, or homology-directed repair, or using non-homologous end joining. Okay, and in the process of doing such, what's going to happen is when you repair that little nicked DNA, you're going to insert a mutation. You're going to have a little bit of an insertion, a little bit of a deletion, a little bit of a SNP here and there. You're going to have a scar, and that scar essentially constitutes a mutation exactly at the location of cleavage. And that essentially is the underpinning of genome editing. You're editing the sequence. You're changing the content of that sequence at the cleavage site. That's genome editing 1.0. And in other ways, if you don't use this quick and dirty DNA repair type alternative, you can use homology-directed repair, whereby you can provision a template to instruct the cell as to how to repair its DNA. And really, by design, you can engineer the nature of the mutation that you want, either an insertion, a deletion, or a customized designer mutation. Okay, there's other ways to do that, and I'm not going to waste your time discussing this too much. That's the underpinning of CRISPR-based genome editing. Now, this is genome editing 2.0, and kind of disregard the formatting challenges here. So Cas9 in general is associated with genome editing, as just before mentioned, to delete, insert, or knock out a DNA sequence of interest. That's using the canonical Cas9. That's like CRISPR circa 2013. It's so long ago in the age of CRISPR that it's almost obsolete, not quite yet. And if you move on to 2014 and 2015, there's other ways to use Cas9. It's a deactivated version of Cas9 in which the cleavage domains have been inactivated. And now instead of cleaving DNA, you just target DNA and bind DNA specifically at a location of interest. And what you can do is you can fuse Cas9 to transcriptional activators to induce transcription. You can fuse Cas9 to transcriptional repressors to repress transcription. You can fuse Cas9 with any effector domain of interest to recruit and locally deliver an effector domain of interest to a sequence of interest. You can fuse Cas9 to fluorophores and imaging molecules and visible molecules to do live imaging in single cells to visualize the location and or the state of your chromatin in our sequence. And then last but not least, actually the Charlie Gersbach lab was very instrumental in showing that you can use versions of Cas9 fused with acetyltransferases or methylases to really alter the epigenetic state of a sequence of interest and not just rewrite the genome using CRISPR, but actually rewrite the epigenome as well. So you can rewrite the transcriptome, rewrite the epigenome, and other things. Ergo, CRISPR 2.0 and genome editing 2.0. Now, the rise of this molecular scalpel technology here is really inherent to the many advantages that it provides and construes. It's very programmable, it's very specific, it's transferable to any species of interest. I think now we have hundreds of cells in which it's been done. It's very efficient relative to some incumbents. It's very precise in terms of cleavage. It's affordable, I think, in terms of design and in terms of implementation, in terms of reagents. That has changed the game, arguably. It's very quick and easy to implement. Even like a random student at NC State can do that, let alone Duke. And it's very multiplexable in the sense that you can use multiple guides concurrently or iteratively. And also sometimes you can use multiple different scalpels that are orthogonal to do different things at the same time. So you could think of upregulating certain pathways and at the same time using an orthogonal system to downregulate other pathways and really tinker with biological processes within one cell at the same time. Now, however promising as CRISPR has been, this is still a 2-year-old child, golden child, charming, promising, high potential, but like any 2-year-old technology, it needs improvements. And Cas9 is still somewhat large, difficult to package in retroviruses. We may hear about that from Brian. You have some sequence-dependent targeting that's biased. We may or may not hear about this from Charlie. There's some concerns about off-target cleavage, but they've been mostly overdone, but they are being addressed from a technical standpoint. And then one challenge is, some sites cut better than others, and that's the part of the mystery of DNA that is yet to be unraveled. And moving forward, there's a lot of efforts ongoing right now to really optimize guard RNA selection, design, and composition, expanding the array of enzymes available to us, expanding the array of sequences available to us, and then really trying to move towards, can we predict what the genome editing outcome is going to be at a site of interest? So that technology is moving along quite nicely, quite well. Now, for the sake of business, one thing that's interesting is that the CRISPR craze, however much advertised and championed by the scientists, right? And we see the rate at which papers are coming out. We see the rate at which the literature is being cited. We see how visible it is in all kinds of high impact factor journals and whatnot. Regardless of how much of an impact it's had at a scientific level, from a media standpoint, it's even been more compelling, arguably, right? And we see a lot of people and a lot of different highly visible media cover CRISPR. He made the cover of The Economist in one of the issues about four issues ago. 2 of the time 100 most influential people in the world are now CRISPR scientists and pioneers. And there's stories in Forbes, New York Times, NPR. It's all over YouTube and the Twitterati and whatnot. And it's been really, really compelling and interesting. There's been some good and some bad coverage. We'll get into that, I'm sure, in the panel. But regardless of the interest from the scientists and the media, who both have a propensity to some extent to overhype their own work, I think some of the most interesting and compelling story about the CRISPR craze lies with the business. And it's been very impressive, if not unprecedented, to see at the same time different types of business segments get into the CRISPR game very, very quickly. There's been a lot of talk, and you'll see, and the illustrations we get from our fellow speakers today here, how much of an impact it's making in medicine for antiviral alternatives, or for gene therapy alternatives. You'll hear from one of each topic from each speaker. But notwithstanding the potential in medicine and next generation medicine, there's a lot of potential already for ag, for biotech, and for food. Okay, so if you want to talk about how much CRISPR enhanced products you may have consumed the last 5 years, ask me the question in about 10 minutes. And then at the same time, what's compelling is not just the breadth of types of business segments that have gathered CRISPR interest from a strategic standpoint, but also the different types of companies that have seen the pie big enough to get into the game. And we have some Fortune 10 companies, we have a lot of Fortune 500 companies, obviously, but we also have mid-cap companies, we have startup companies, we have VCs investing hundreds of billions of dollars in those technologies, and we even have not-for-profit companies get into the same game at the same time on the same innovation and same technology. That's not very common, and I think that's noteworthy. And I think a few years from now we'll hear about cases of how the CRISPR craze took off. Now, that being said, there's concerns about IP and ethics and GMO versus non-GMO and regulatory path, which I'm sure we'll discuss during the panel. And already there's a lot of applications that have occurred outside of medicine and bacteria. That's just the tip of the iceberg. So you have food starter cultures enhanced with CRISPR. You can use CRISPR to genotype bacteria of interest. And there's a whole slew of animal and industrial applications that are already in motion that encompass a broad range of the food supply chain. So last but not least, we heard about what it takes to be successful from an innovation standpoint. And as a food scientist, one of my degrees I've had, it's all about putting the right ingredients together. You need different things to come and get aligned at the right time, the right place, with the right topic, the right business opportunities. And as long as you put all those different things together the right way, you can expect nothing else than success and a masterpiece. But it's a lot harder to do in reality than it is to push forward. So I'm going to stay quiet from here on. I think we'll hold off questions until the Q&A part of the topic, and I will leave the floor to the real scientists in the room.
Thank you, Dr. Barrangou. So let me introduce our next speaker, Dr. Cullen. So Dr. Cullen is a prolific publisher, is all I can say. You can read his bio in here, but he's published over 300 research papers. He's the director for the Duke Center for Virology and a professor of molecular genetics and microbiology. His primary area of research is in viral microRNA and CRISPR-Cas for editing viral DNA genomes. Dr. Cullen?
Okay, thanks. So are these my slides? We'll see, I guess, if I press this. So my brief from Rodolphe was to talk about the use of CRISPR-Cas as a potential therapeutic intervention in important viral diseases of humans. And that's not a huge leap of the imagination, given that the CRISPR-Cas system evolved in bacteria as an adaptive immune response to prevent infections by viruses, specifically bacteriophage, which are DNA, generally speaking, DNA genome-encoded viruses. So CRISPR-Cas is an RNA-guided DNA endonuclease, as Rodolphe said, and so we're restricted to a significant degree to DNA viruses. So we work on two. One is hepatitis B virus, which is a very important infection that has over 300 million infected individuals worldwide, but I'm not really going to talk about that because of time limitations. Instead, I'm going to discuss human papillomavirus. Human papillomavirus is a small DNA virus that causes a number of important diseases. It's very tightly linked to cervical cancer, but it's also linked to other sexually transmitted cancers, particularly head and neck cancer, where we get about 21,000 cases annually in this country, and anal cancer, where we get about 6,000 cases annually. And these are almost invariably caused by one type of papillomavirus, which is HPV-16. There are actually over 110 human papillomaviruses, many of which are associated with benign warts or other inconvenient but not particularly dangerous manifestations. But a couple of viruses cause cervical cancer, and then you have this one virus, HPV-16, that causes head and neck and anal. Now, those respond reasonably well to chemoradiation therapy, so you get about a 70 to 75% cure rate, but then the 25% or so that are not cured by the more traditional cancer interventions are in deep trouble because head and neck cancer, there's not a lot of room for whacking big pieces of tissue out of the back of your throat without causing severe damage. And so salvage therapy is very deleterious in that case. And anal cancer, you can operate to remove it, but then you lose your anus, which is, means you have a colostomy bag around with you the rest of your life. So we believe this is an interesting little niche for targeting because these cancers are very deleterious, and I think this is the kind of thing that where we could get potentially FDA approval to use a CRISPR-Cas approach to treatment more readily than perhaps hepatitis B virus, which only actually kills about 20% of the patients that it infects with a long time lag. So we're going to target HPV-18 in this case for convenience reasons in a cervical carcinoma cell line with CRISPR-Cas. So HPV normally replicates as an episome, and it has a series of genes. Some are what are called early genes marked with an E, and then late genes marked with an L. The virus replicates the DNA in completely differentiated basal cell layers underneath the skin or the mucous membrane, and as the cells migrate towards the surface of the skin, the early genes are switched off and the late capsid proteins are switched on, and then where the wart is, you're going to be releasing DNA particles into the outside surface. So some are born by, get into skin lesions, and that's how you get a wart, but then a lot of these viruses are actually transmitted by mucous membranes, so particularly in the genitalia. So normally, this can be a little annoying, but it isn't such a big deal. But for some of these viruses, they have the potential to actually cause cancer. And so in invasive cervical cancer, what you get is an integration event where this normally circular DNA that replicates as a circle in the nucleus, integrates into the human genome and disrupts the E2 gene. Now, the E2 gene normally keeps E6 and E7 turned very low. And when E2 is disrupted by integration such that the open reading frame is broken, E6 and E7 are turned up very high. And what E6 does is it turns off a protein called P53, which is a host cell response gene that determines that DNA damage has occurred. And this is called the guardian of the genome because it's the single most important factor in telling a cell to commit suicide if the cell is growing aberrantly. There's another defense mechanism called the retinoblastoma protein, and that's because it was originally identified as a gene lost in retinoblastoma tumors, which are inherited tumors in the eye of some children. And again, the retinoblastoma protein is able to turn off replication of the cell and prevent a cancer cell from growing. In normal cancers, these are often disrupted, but in HPV-induced cancers, they're actually turned off by E6, turns off P53, and E7 turns off RB. So that these host cell response elements that would make a cancer cell kill itself rather than continue to grow are no longer effective. So we have designed some CRISPR-Cas guide RNAs to target these genes. And here's an indicator assay. And what we do is we have an epitope tag that allows us to measure the presence of a protein, and then green fluorescent protein, which is, as its name says, fluoresces green. And we put a target between these two, and then we can introduce into cells by co-transfection the Cas9 protein from an expression vector and a single guide RNA or sgRNA specific for this inserted target, which is derived from HPV E6 or E7 gene. And we ask, do they work? And we can show that we get a very strong knockdown when we have these guides present for either E6 or E7 in this artificial sort of setting. And you can see here that if we have a protein that is got an E6 target, when we have an E6 specific guide RNA, the expression of the target goes away compared to the control. And here we have E7 targets. You can see the protein goes away compared to the control. So this just shows that at least in a cell, in some kind of artificial setting, that these work. More importantly, then, we can introduce them into an actual cervical carcinoma cell line. So these are cells that are transformed by HPV. They're recovered as a tumor from a woman, and you can culture them in a tissue culture setting. And now we're going to hit E6, and we're going to ask, what do we see? So here is the target, underlined here in red. This is something that Rodolphe didn't really talk about, but it's an invariant part of the target, which isn't part of the RNA recognition element. It's directly recognized by the Cas9 protein itself. It's called PAM. I won't tell you what that stands for, but has to always be there. And the cleavage site, we know, is 3 nucleotides to the left-hand side of this PAM sequence. So we're going to go 3 this way. And if we take these cells and we introduce into the cancer cells the guide RNA and Cas9, and then we recover the DNA by PCR, what we can see is that we get editing events all around the expected target site. And here, for example, we have 34 clones that we've recovered with a single nucleotide insertion, and all of those 34 insertions are 3 nucleotides away from the PAM. And so these are in an open reading frame. They're in a protein open reading frame, and so they'd be expected to disrupt the expression of the protein. We can also do this with the HPV18E7 gene, and here again we see insertions 3 nucleotides away from the PAM, 1 nucleotide or 2. We also see deletions, and we also see this one interesting large insertion where a little piece of human chromosome 8 has wandered away and decided to integrate itself into the HPV18 genome. So this is all very well and good. We are editing, we are destroying these genes. Do we actually, is this doing any good for the purpose of the exercise? So I already told you that E7 targets RB and E6 targets P53, and in both cases, it leads to the destruction of these host defense mechanisms. And so we predict that if E6 is disrupted, P53 should turn on, and if E7 is disrupted, then RB should turn on because it's no longer being degraded. So what we have here then is exactly that experiment. So here's E6 being targeted. And so, as I told you, E6 normally degrades P53. So if we target E6, we should turn on P53. There it is. P53 turns on downstream effectors. So it induces two kinds of effectors. It induces pro-apoptotic proteins that tell the cell to commit suicide. It also induces cell cycle arrest proteins. One of the most prominent of these is a protein called P21, which is frequently disrupted in cancer. And you can see that P21, which is transcriptionally activated by 53, is also turned on. What you can see down here, and it's not quite as striking, is RB is being activated. And you can see there's more RB here, and it's actually quantitated underneath here. There's a slight complication with these kinds of assays because when you turn on RB and P53, you actually start to kill the cells. And so you can see that the Cas9 protein itself, which is designated here as anti-flag, is going down. And here it's going down even more when we're hitting the E6 and E7 gene. And that's because there are just a lot fewer cells in the culture. And so this increase in expression has to be sort of factored by the fact that we're looking at half as much gene expression in the transduced cells. I won't talk about the data on the right, but it just confirms the specificity of what we're seeing. So I told you if E6 turns on, if E6 or E7 is turned off, now we should activate P53 or RB. The cells should stop growing and or die. So here we have a culture where we have cells that we've transduced with something that's specific for E7. Here's one that's specific for E6. And here is one that's the Cas9 protein with a guide that's not specific for anything to do with the virus. And what you can see is the cells that don't have an HPV-specific guide continue to grow happily. The ones that have a guide specific for HPV E6 or E7 die. And what we can see is that they actually start to arrest, and I don't want to go through the details of this assay, but simply put, cells go through a cell cycle where they go through G1, then they go into S, which is DNA synthesis, then they go into a resting phase called G2, and then they go into a phase called M, or mitosis, where they actually divide. And so what you can see is that when we have E6 or E7 turned on, the number of cells undergoing DNA synthesis has dropped substantially, and the number of cells arrested in the G1 phase of the cell cycle has gone up substantially. So we're seeing both cell death and cell cycle arrest. So we can actually do this then with a lentiviral vector, where we can deliver the CRISPR-Cas system in its entirety using a single vector, which is based on HIV-1. And this is an experiment where we're transducing with either the blank vector or a vector with a non-specific guide RNA or with guides specific for E6 or E7. And then we're looking at the ability of these cells to grow out. We're hitting with a multiplicity of infection of 2. And so we would predict that we would infect 90% of the cells on average, and about 10% of the cells would not be infected at this titer. And what you in fact see is that 10 days out, we're about 10-fold more cells in the controls than in the ones with E6 or E7 transduction. And remember, 10% of the cells would be predicted not to get anything, and those would continue to grow, and all of the other ones have died. So you see quite a dramatic effect here. But there are still cells here that are growing, so we can also hit the cells with a much higher titer. So we can hit the cells with a multiplicity of infection of 30 per cell in this culture system. And if you do that, you actually start to cause a little bit of just cytopathic effect just from the fact that there are so many lentiviruses infecting the cell, even though they're replication incompetent vectors. But you see a bit of a drop relative to your mock. But now you see that the ones that got the E6 or E7 guide are completely toasted relative to the non-specific guide. So here we, okay. So what we could do then is we can cleave the target in these cells, and we can recapitulate the expected phenotypes. And so this is basically the ending point. How do we deliver this in vivo? The CRISPR-Cas, the Cas9 protein that Rodolphe and everybody else has worked with the most is from a bacteria called Streptococcus pyogenes. We've actually been working a great deal with a protein from Staph aureus, which is a lot smaller, and that allows us to put it into an adeno-associated virus vector within the packaging limit of that vector, which is 4,800 base pairs. SPY-Cas9 is 4.2 kb, so we can't fit it in. But we now have these vectors. We've packaged them. We have them in mice. We can get titers of up to the 10 to the 14 viral particles per ml with the AAV system. When you can get 10 to the 14, which is a very big number, into a 1 ml, you can actually transduce whole livers, you can transduce whole organs. So I think that this particular approach in terms of being able to deliver the Cas9 protein and guide RNAs, all within the context of an AAV-type vector, is very, very feasible. And indeed, there's been one paper from the Fang Zhang's laboratory using exactly the same protein, which they came over across somewhat independently from us, and showing that they could get extremely successful transduction of the liver, so that they were able to knock down LDL production in the mouse by 90%, and basically ablate the expression of a particular gene in the liver. So this work was done in my lab at Duke, and particularly was done by, which is interesting because it's his slide set, so his name isn't up here, but Matt Kennedy is the postdoc who did this work, working particularly with Anand Kanaparty, Adam Meffert, and Hal Bogut in my lab. And we've had some collaborations with the Ray Shinazi's laboratory at Emory and the Castlin lab at Duke, who's interested in the application in a cancer setting. We have some of this work was funded by Hoffman LaRoche. Thank you very much.
Thank you, Dr. Cullen.